The history of human evolution is often presented as a branching tree, filled with one species replacing another in rapid succession. Every few 100,000 years, a new name appears, a new skull is discovered, and another branch is added to the family tree. Homo habilis gives way to Homo ergaster, which gives way to Homo erectus, while Homo rudolfensis, Homo georgicus, and Homo naledi each occupy their own distinct places along the journey towards modern humans. It is an elegant picture, but elegance is not the same as accuracy. Sometimes the most important scientific discoveries do not add another branch to the tree. Instead, they force us to wonder whether some of those branches ever existed at all. A remarkable study, published in 2026, has introduced exactly that kind of question. Using a technique that would have seemed impossible only a few years earlier, scientists extracted ancient proteins preserved inside the enamel of fossil teeth belonging to Homo naledi, the mysterious small-brained human discovered deep inside South Africa's Rising Star Cave. The purpose was straightforward. By identifying proteins linked to the X and Y chromosomes, the researchers hoped to determine the biological sex of the individuals represented by the fossils. The expectation was simple. Some fossils would prove female, while others would prove male. Instead, the result surprised everyone involved. Every tested individual lacked the protein normally associated with the Y chromosome. Every one of the 20 individuals examined appeared to be female. The finding immediately created one of the most intriguing mysteries in modern paleoanthropology. The research itself does not establish that every Homo naledi individual ever discovered was female. Instead, it demonstrates that every individual tested from this unusually large sample lacked the male-specific enamel protein marker. The researchers repeated the work using additional teeth, examined fossils previously considered likely males because of their larger size, and even had an independent laboratory perform the same analyses. The outcome remained unchanged. Every tested specimen appeared female. Rather than answering one question, the study created several entirely new ones. The first mystery concerns the cave itself. Why would only females appear inside the Rising Star chamber? If these individuals entered the cave through some form of intentional activity, why were males apparently absent? The research team suggested that males and females could have received different treatment after death. Other scientists questioned whether there could be an unusual genetic explanation involving the enamel proteins themselves. Still others proposed that the cave simply preserves an incomplete picture of a once much larger population. None of these explanations has yet resolved the puzzle. The fossils have simply become more mysterious than they already were. What makes this discovery particularly significant is that it reaches far beyond Homo naledi. For well over half a century, paleoanthropologists have relied almost entirely on differences in skull shape, jaw structure, brain size, and facial proportions when assigning fragmentary fossils to different species. A robust skull often becomes one species. A slightly smaller skull becomes another. A wider face receives one scientific name, while a narrower face receives another. Those decisions are made because complete fossil populations almost never exist. Scientists usually work with isolated skulls, partial jaws, scattered teeth, and incomplete skeletons separated by hundreds of thousands of years and enormous geographic distances. That reality creates an unavoidable problem. If researchers cannot confidently identify males and females within extinct populations, how can they know whether anatomical differences represent entirely separate species or simply ordinary biological variation? Living primates demonstrate that males and females frequently differ dramatically in body size, skull robustness, muscle attachments, facial proportions, and overall appearance while remaining members of exactly the same species. Gorillas provide perhaps the most dramatic example, with dominant males weighing nearly twice as much as females. Baboons, orangutans, and many other primates display similarly impressive differences. Fossil humans almost certainly displayed some degree of sexual dimorphism as well, yet measuring its true extent has always remained extraordinarily difficult. The Homo naledi study suddenly shines a spotlight on this long-standing problem. If 20 individuals that collectively span much of the known anatomical variation of the species all prove female, then scientists still have almost no direct knowledge of what confirmed males actually looked like. The larger skulls that once seemed obvious candidates for males apparently are not males at all. That realization forces a simple but profound question. If the variation within Homo naledi females alone is already substantial, how much larger was the total range once males are added to the picture? That question becomes especially interesting when attention turns toward Homo habilis. Ever since its description in the early 1960s, Homo habilis has occupied an unusual position in human evolution. Its brain size generally ranges from about 500 to 600 cubic centimeters, larger than most Australopithecines, but noticeably smaller than later humans. Its face is flatter than earlier forms, its brain case larger, and many aspects of its anatomy place it comfortably within the human lineage. Yet the fossils assigned to Homo habilis vary considerably. Some skulls appear small and lightly built. Others are noticeably larger and more robust. Rather than representing ordinary variation within one population, many of these differences gradually became the basis for naming entirely different species. One of the most famous examples is KNM-ER 1813, discovered near Lake Turkana. With a brain measuring roughly 500 cubic centimeters, it has traditionally represented a classic example of Homo habilis. Not far away, other fossils, including KNM-ER 3733 and KNM-ER 3883, possess substantially larger brain cases approaching 850 cubic centimeters, while sharing many other anatomical characteristics. These larger fossils became identified as Homo ergaster. Meanwhile, the larger-faced KNM-ER 1470 received the separate name Homo rudolfensis. Instead of one variable species, the fossil record became divided into several closely related ones. The boundaries separating these groups, however, have never been entirely convincing. Brain size alone forms a remarkably continuous progression rather than distinct clusters. Facial proportions overlap. Cranial architecture shares many important characteristics across specimens. Geographic distributions overlap almost completely. Time periods overlap as well. Instead of one species disappearing before another arrives, these populations often appear to exist simultaneously within the same East African landscapes. If Homo naledi demonstrates anything, it is that appearance alone does not necessarily reveal biological sex. The larger individuals, once expected to represent males, apparently do not. That realization naturally invites a broader question. Could similar assumptions have influenced the interpretation of much older fossils? This possibility becomes even more intriguing when considering the famous Turkana boy skeleton. Discovered in 1984, Turkana boy represents one of the most complete early human skeletons ever found. Assigned to Homo ergaster, the skeleton possesses unusually long legs, a relatively tall body, and an adolescent brain size, estimated at roughly 750 cubic centimeters. Because complete skeletons from this period are exceptionally rare, Turkana boy has often served as the standard model for what Homo ergaster looked like. Yet Turkana boy also stands somewhat alone. Very few other fossils from the same period preserve comparable body proportions. His skeleton has been interpreted as representing the emergence of the tall, modern human body plan in Africa, but his skull still shares numerous characteristics with smaller-brained African fossils traditionally assigned to Homo habilis. Rather than representing an entirely separate evolutionary stage, Turkana boy also fits within a broader continuum extending across East Africa during this period. Similar patterns appear outside Africa. The fossils discovered at Dmanisi in Georgia demonstrate remarkable variation despite belonging to a single population living at approximately the same time. Brain sizes differ substantially, faces differ noticeably. Some skulls appear much more robust than others. Yet because these individuals were recovered together from the same archaeological setting, many researchers interpret them as members of one highly variable population rather than multiple species. Dmanisi serves as a powerful reminder that isolated fossils recovered from different locations naturally exaggerate apparent diversity. The comparison with Homo naledi becomes impossible to ignore. Both populations possess relatively small brains compared with later humans. Both exhibit unexpectedly human body proportions, combined with more primitive cranial features. Both demonstrate anatomical variation that initially encouraged researchers to divide fossils into separate categories. Now, with the surprising discovery that every tested Homo naledi individual appears female, one of the key assumptions underlying those interpretations has become far less secure. If future discoveries eventually identify confirmed male Homo naledi fossils, those individuals could prove substantially larger than any currently known specimen. They could possess more robust skulls, larger faces, and noticeably greater brain volumes while remaining members of exactly the same species. Such a discovery would significantly expand the recognized limits of anatomical variation within one human population. This possibility naturally raises broader questions about the entire early human fossil record. How many named species actually represent genuine biological species, and how many simply reflect incomplete sampling of populations whose males and females differed more dramatically than previously recognized? The answer remains unknown, but the question itself has become increasingly difficult to ignore. If fewer biological species existed than currently recognized, then the familiar picture of human evolution changes in important ways. Rather than a crowded evolutionary landscape containing numerous closely related species replacing one another across Africa, the fossil record instead begins to resemble a smaller number of long-lasting lineages displaying considerable variation through time and between the sexes. Such a model does not eliminate evolutionary change. Instead, it changes the scale at which that change occurred. This discussion also reaches one of the most important debates surrounding human origins: the evolution of larger brains. The traditional narrative proposes that progressively larger-brained humans evolved within Africa before dispersing into Eurasia. Species such as Homo ergaster occupy a central role within that interpretation because they represent an apparent intermediate stage between smaller-brained Homo habilis and later humans, possessing cranial capacities approaching or exceeding 1,000 cubic centimeters. If, however, Homo ergaster ultimately proves to represent the larger-bodied sex of a broader Homo habilis population rather than a separate evolutionary stage, that straightforward progression becomes far less certain. Instead of documenting one species replacing another through steadily increasing brain size, the African fossil record would document one highly variable lineage whose members occupied a broad range of body sizes and cranial capacities. That alone would not overturn the evidence that larger-brained humans eventually appeared. Asian Homo erectus fossils unquestionably include individuals with much larger brains than classic Homo habilis. African fossils such as Bodo, Buya, and Dhaka likewise demonstrate substantial cranial enlargement during later periods. Those discoveries remain firmly established regardless of how earlier fossils are classified. What changes is the evolutionary pathway connecting these populations. Rather than viewing Homo ergaster as an obvious transitional species leading directly toward larger-brained humans, scientists would instead need to reconsider which populations actually contributed to later evolutionary developments and which simply survived alongside them. The evolutionary tree becomes less linear, more complex, and perhaps surprisingly simpler in terms of the number of biological species involved. That is why the new Homo naledi protein study deserves attention far beyond one South African cave. Its greatest importance does not lie in proving an extraordinary conclusion. Its importance lies in exposing a hidden assumption that has quietly shaped interpretations of fossil humans for generations. Researchers have often believed they understood the biological sex of extinct humans simply by comparing skull size and skeletal robustness. The enamel proteins now suggest that those assumptions deserve careful re-examination. Scientific progress often begins not with answers, but with questions that nobody expected to ask. 20 ancient individuals lying deep beneath the South African landscape have introduced precisely that kind of question. Their small brains once seemed to reinforce familiar ideas about primitive humans. Instead, they have become the source of one of the most unexpected mysteries in recent paleoanthropology. If every tested individual truly represents a female, where are the males? What did they look like? how much anatomical variation existed within this population. And if our understanding of one species can change so dramatically through a single new analytical technique, what other assumptions about the early human fossil record remain waiting to be challenged? Those questions remain unanswered, and that is exactly what makes this discovery so fascinating. The history of human evolution has never been written in stone, even when it is built from stone-like fossils. Every new technique reveals details that previous generations could not see. Ancient proteins have now opened a completely new window into the biology of extinct humans. And through that window, we are beginning to glimpse a picture that is simultaneously simpler and more mysterious than the one we inherited. Whether Homo naledi, Homo habilis, Homo rudolfensis, Homo ergaster, and Homo georgicus ultimately represent fewer biological species than currently recognized remains an open scientific question. What is no longer in doubt is that the assumptions guiding those classifications are being tested in entirely new ways, and the answers promise to reshape one of the most enduring mysteries in the story of our own origins. Thank you for watching. Every fossil discovery adds another piece to the puzzle, but every new scientific technique has the potential to change the shape of the puzzle itself. The story of human origins continues to evolve with every new discovery, and some of the biggest surprises still lie hidden inside fossils that have rested silently beneath the earth for hundreds of thousands of years. If you enjoyed this exploration of one of the newest mysteries in paleoanthropology, please consider subscribing and join me next time as we continue searching for the forgotten chapters of our ancient past.